Hello students, welcome back to a new video of Sunanda's tutorial. Today we will discuss the secondary growth in dicot stem of higher plants. So what is growth? Growth either occur by length or by thickness. The growth in length of the plant axis is known as primary growth and it is developed or produced by the primary tissues, and the primary plant bodies are developed by apical meristem. The primary growth of the plant body occur by apical meristem, either by shoot apical meristem or root apical meristem. Shoot apical meristem present at the terminal or apex part of the shoot, and root apical meristem is present in the terminal or apical part of the root. Increase in girth or thickness of the axis is called secondary growth. The tissue formed during secondary growth are called secondary tissues. Sometimes, as in monocot and pteridophytes, does not grow in thickness by cambial activity. So, secondary growth is absent in case of monocot and pteridophytes. However, gymnosperm shows secondary growth. Then, angiosperm obviously show secondary growth. Secondary tissues may be of two types: the vascular tissue that developed by the true cambium, the cork, and the phelloderm, which is formed by phellogen or cork cambium. In a typical dicot stem, secondary growth starts in the intra and extrastellar region. In the intra and extrastellar region. So it is the basic points about the secondary growth. Let us discuss this in details. So the secondary growth takes place by the following steps. One is formation of cambial ring. Second step, activity of cambial ring, how they divide, how they act. Then the secondary vascular tissue formation and formation of periderm. So this is the four-step process of secondary growth in dicot stem. One is step 1, formation of cambial ring. Step 2, activity of cambium ring. Third step, secondary vascular tissue formation. Fourth one, formation of periderm. So it is the overall stages of secondary growth in woody stem. So first of all, the step 1, formation of cambial ring. Formation of cambial ring, step 1. The cambium is present in the primary vascular bundle is known as fascicular cambium. What is fascicular cambium? See, this is a vascular bundle. I have already told you in my previous classes that it is vascular bundles present in the stem. Xylem and phloem present togetherly in dicot stem. So it is known as conjoint type of vascular bundle. So between this xylem and phloem, a cambium is present. From that cambium, the secondary growth will occur. So this cambium is known as fascicular cambium or vascular cambium. It is primary in origin. It is present between xylem and phloem during their primary growth. Before secondary growth starts, the parenchymatous cell of the medullary rays, which are in line with fascicular cambium, becomes meristematic and form interfascicular cambium. So these are cambium 1, 2, 3, 4 and 5. These are cambium or fascicular cambium. During secondary growth, these parts are known as medullary rays part. Between vascular bundles, medullary rays are present. These parenchymatous tissue are called as medullary rays. So they become meristematic and they join with each other like this. So this cambium join with each other by forming interfascicular cambium. So at last, a ring is formed. The interfascicular cambium extend laterally and join with fascicular cambium forms a continuous cambial ring. The cambial cells are of two kinds, namely fusiform initial and ray initial. The fusiform initial produces secondary tissue. Ray initial gives rise to ray cell and xylem and phloem. So finally, cambium ring is formed, which is your first step, formation of cambium ring. And I have showed you that how this cambial ring is formed. Next step, activity of cambium ring. After the formation of cambium ring, what type of activity they show? What type of activity was shown by this cambium ring? The cambium ring cuts or divide vascular tissues on either side. The cells that are produced outward become differentiated into secondary phloem, and the cells that are formed inward become differentiated into secondary xylem. That means here it is the cambium ring. It produces secondary phloem towards the outside and secondary xylem towards the inside. Due to the formation of secondary xylem, the primary xylem masses and pushes towards the center. Due to formation of secondary phloem, primary phloem masses and push outwards. So in this way, cambium ring divide and form the secondary phloem towards the outside and secondary xylem towards inside. Then secondary vascular tissues. Secondary vascular tissues are secondary xylem and secondary phloem. They are formed by the fusiform initial of vascular cambium. The secondary xylem is known as wood. It is very important. The secondary xylem is known as wood. The cambium ring cuts off new cells on inner side and gradually modified into xylary element called secondary xylem. The tissue serves many important functions such as conduction of water, nutrient, mechanical support, etc. Cambial cell divide tangentially, producing secondary phloem element towards outside of it. So this cambial cell or cambium ring divide tangentially. That means suppose it is a cambium, it divides like this transversely. It divides transversely and form towards phloem towards outside and xylem towards inside. Secondary phloem is also known as bast and consists of sieve tube, companion cell, phloem fiber and bast fiber and phloem parenchyma. So these are four components of phloem. One is sieve tube, one is companion cell, which is always associated with sieve tubes. One is phloem fiber and the last one is phloem parenchyma. Normally, the amount of secondary phloem is lesser than the amount of secondary xylem. Usually, the primary phloem becomes crushed and functionless, and the secondary phloem performs all physiological activities for sufficiently long period of time. So, it is all about secondary vascular tissue formation. Then formation of periderm. What is periderm? Due to continued formation of secondary tissue, the older stem, the epidermis gets stretched and ultimately tends to rupture, followed by death of epidermal cell and the outer tissue, and a new protective layer is developed called periderm. So during the secondary growth, the primary outer layer or epidermis gets degraded or ultimately ruptured and leads to death, and a new protective layer is formed in the place of the primary epidermal layer. The formation of periderm is a common phenomenon in stem of gymnosperm that increase in thickness by the secondary growth. So structurally, periderm consists of three parts. The periderm is produced new lateral meristem called phellogen or cork cambium. The layer cuts off by phellogen on outer side called phellem or cork, and the cells cut off phellogen towards inner side called phelloderm, and it is known as secondary cortex. The periderm appears on the surface of those plant parts that possess continuous increase in thickness by secondary growth. So, I have told you one thing, that it is a secondary outer layer of the plant. What is periderm? That means what is the structure of periderm? It is consist of three parts structurally. One is cork cambium. The periderm produce new lateral meristem called as cork cambium. This cork cambium layer cut or divide and form phellem or cork towards outside and phelloderm towards inside. So the phellogen layer or cork cambium, which is a type of secondary cambium meristematic tissue, they form cork or phellem towards outer side, cork or phellem towards outer side, and phelloderm towards inner side. And these three layers togetherly consist of the periderm or secondary outer layer, secondary epidermis, you can say. Next, annual rings or growth ring. What is annual rings or growth ring? The secondary xylem in the stem of perennial plants commonly consist of concentric layers. Concentric layers means like this. So these are concentric layers, each one of which represent a seasonal increment in transverse section of the axis of the plant axis. These layers appear as a rings and called annual rings or growth rings. Annual ring or growth ring. The wood developed in the summer or spring season is called spring wood or early wood, and the wood formed in the winter or autumn called autumn wood. So at two times of a year, this wood developed. One is summer or spring called as spring wood, another one in autumn called autumn wood or late wood. The line of demarcation is quite conspicuous between late wood and of one year and the early wood of the next year. An annual ring therefore consists of two parts, one inner layer or early wood and outer layer called late wood, which is formed in the autumn. So it is the diagram showing the annual rings in an old stem. Then dendrochronology. What is dendrochronology? Each annual ring corresponds to 1 year of growth. On the basis of this ring, age of a particular plant can be calculated easily. So the determination of the age of a tree by counting the annual rings is known as dendrochronology. So in a year, at two times the growth of the plant occur, at autumn and in spring. So at two times the growth occur, and so two rings formed in a year. So by just counting those rings, you can tell that easily tell the age of the plant. So to counting the age of a plant by simply counting the annual rings is known as dendrochronology. Then what is stylosis? In many plants, the walls of xylem vessels produces a balloon-like outgrowth into the lumen of the vessel called stylosis. It is very important for you from your examination point of view. Then what is sapwood and what is heartwood? The outer region of older tree consists of recently formed xylem element that is known as sapwood or alburnum. This is of light color and contains some living cells, also in association with vessel and fiber. This part of the stem performs physiological activity such as conduction of water, nutrients, because they are recently formed and living. The central region of older tree was formed earlier and filled up with tannins, resins, gums and other substances which make it hard and durable. It is called hardwood. hardwood or duramen. Softwood or the outer part which is recently formed and contains many living part is known as sapwood or alburnum, and the central part of the stem or older tree trunk is known as hardwood or duramen. It looks black. Why? Because the presence of various substance like tannin, resin, gums. Usually vessels remains clogged with tyloses in hardwood. The function of hardwood is no longer in conduction. It gives only the mechanical support to the stem, and the conduction occur in sapwood, and the mechanical support is provided by the hardwood. The sapwood changes into hardwood very gradually. The proportion of sapwood and hardwood is highly variable in different species. So, it is the transverse section of stem showing hardwood and sapwood. So, it is the black part is known as the hardwood and the lighter part around it is known as sapwood. Next, bark. The term bark is commonly applied to all tissues outside the vascular cambium of the stem in either primary or secondary state of growth. In this way, bark includes primary phloem, cortex in stem with primary tissues only, and primary and secondary phloem, cortex and periderm in stem with secondary tissues. So the next is what is lenticel? Lenticel is usually the periderm or the secondary epidermis of most of the plant, contains the area with loosely arranged cells have been found. These possess more or less raised or corky spots where underneath tissue break with break through the epidermis. Such areas are universally found in stem of the woody plants. These broken areas are called lenticels. Woos in 1955 defined a lenticel as small portion of the periderm where activity of phellogen. What is phellogen? That is cork cambium. So the activity of phellogen is more than elsewhere, and cork cell produced by it are loosely arranged and possess numerous intercellular spaces. The lenticels perform the function of exchange of gases during night or when the stomata are closed. So it is the lenticel which is generally found in woody plants. So it is all about your secondary growth in dicot stems of higher plants. In my next class, I will teach you about the secondary growth in roots of dicot plants. So stay tuned and enjoy my videos. Thank you.